In the first part of the video on our visit to Genethon, a leading laboratory in designing gene therapies for rare diseases, we explored the basic unit of all living things, the cell. When explaining their function, scientists often refer to cells as miniature factories with storage, transportation, and communication departments, as everything the body needs to function is produced there. For example, the cell's nucleus, or the CEO's office, contains our DNA. A particular fragment of the DNA stores the instructions needed to produce a protein, such as the clotting factor protein. This fragment is known as the gene. Each of our cells is constantly dividing and replicating into two daughter cells. This process of division is called mitosis, but before it happens, the cell DNA has to be copied so that each new cell receives a full set of instructions on how to function. Each time mitosis occurs, a mutation in the sequencing of the DNA base pairs adenine, thymine, cytosine, and guanine can take place. Most often, the mutation is insignificant, but in other cases, it could lead to the creation of a defective gene. As explained at Genethon, genetic disorders, such as hemophilia, are caused by a defective gene that prevents the production of a fully functional protein. Therefore, gene therapy involves inserting a normal gene into the cell so that it can make the missing or deficient protein. To transfer the gene, scientists use what is called a vector, or a virus that is rendered harmless, which has the capability to go through the cell wall and into the nucleus and deliver the new gene. Elena Barbin, who holds a postdoc position at Genethon and focuses on developing gene therapy strategies for bleeding disorders, explains the recent achievements and challenges of gene therapy for hemophilia and von Willebrand disease.
Gene therapy for hemophilia is progressing, I can say, really fast, but still there are some challenges we have to take in account. The first one, I would say, is the immune response. So when you inject the viral vector, you can have the immune response for the body against the vector that you infuse or against the therapeutic gene. So now research is really focusing on how to avoid or to escape from the immune response in order to permit the re-injection of the vector, especially if we think about pediatric patients.
Okay. So for hemophilia A, I mean, there it's a little bit more challenging with the size of the gene.
The size of the gene is a problem because the normal gene doesn't fit in a viral vector, but now for the moment we have solved the problem by putting a modified version of the protein. So for this we are okay. The other problem in hemophilia A is that factor VIII is less expressed compared to factor IX and is poorly secreted into the circulation. So the levels that we obtain are lower compared to factor IX. So to have the same efficacy, we need to really increase the dose of the vector with a consequent problem in terms of immune response. Because if we increase the dose, we increase the risk of the response from the immune system. The story is much more complicated for von Willebrand factor because still we have not reached let's say an optimal viral vector to deliver the gene, which is quite big and doesn't fit in a single AAV vector. One of the strategies that we can use is to use two different AAVs to deliver the therapeutic gene. Of course, this is not a super efficient way to do it because you need to have the co-infection of the two viruses in the same cells to have the von Willebrand factor expressed. And another issue is that von Willebrand factor is naturally expressed from endothelial cells, which is the natural biosynthetic site. So the expression and the regulation of the protein is quite complex. So what we need to do is to really find out the way to target the endothelial cells in a way that we can express von Willebrand factor in a proper manner.